Okay, now we will discuss in this part of the chapter about the DNA repair system. So here we will see that this slide is showing what kind of DNA alterations can be present and what kind of damage is there and how that damage is repaired. So it may be spontaneous, the changes in the DNA may be spontaneous, and they may be environmental, they may be beneficial, and they may be harmful. What kind of benefits we can have from the DNA alterations? DNA based sequence, when there are mutations, they are going to provide the genetic variability and survival of the fittest, right? We have heard about this, that we can and the harmful effects of these mutations are that there may be some disability, there may be some poor health conditions, the painful life, and it may result in early death as well, because many of the mutations they results into the development of a cancer, and that cancer is very dangerous and it is very difficult to treat, and even if we can treat it, the quality of the life is very poor. So the most of the DNA damage, it is repaired shortly after it occurs because of the proofreading activity. As I told you earlier, that the proofreading activity of the enzyme, polymerase enzyme, is there 3 to 5 exonuclease activity. For example, if by chance any nucleotide is attached to the wrongly, this nucleotide can be removed by the process of proofreading. And if there is sometimes proofreading cannot correct some of the mistakes, so what will happen, that the repairment of the repairing capability of the repair mechanisms, that will come into effect, and we will discuss that how those defects are corrected. Okay, so there are some types that if there are mutations in the somatic cells, that mutations cannot be transferred from one generation to the other generation. But if there is a mutation in the germline cells, just like sperm and eggs, so they can be passed on from one generation to the other. Spontaneous DNA damage. Every day we are having replications of the DNA in our cells, especially the cells that are responsible for the formation of the blood cells, right? And they keep dividing again and again and again from their bone marrow. Okay, because the blood cells, they are replaced, they are recycled. For example, the RBCs, they recycled every 120 days. Similarly, our immune cells, they are recycled, and that's why we have thousands of spontaneous mutations in our DNA on daily basis. Okay, so there are some types here. Depurination, D, whenever in the biological sciences the word D means removal of something. So here depurination means that removal of either adenine or guanine because of the spontaneous hydrolysis of the glycosidic bond that links it to the deoxyribose. Okay, so DNA in a human cell may lose thousands of purine bases every day. So this is quite normal, and this mechanism is very common in the human cells. And similarly, the deamination, removal of amine group. The amine group that is present in the base, it is altered, and it involves cytosine, adenine or guanine. Changes of the base pairing properties occur as far as that affected base is present. So these three bases, cytosine is the one that is more susceptible to the deamination, and because of the removal of the because of the removal of the amine group, that cytosine is converted into uracil. And here we will see that why uracil is not present in DNA. We will see in a minute that why nature has preferred thymine rather than uracil in the structure of DNA. So in normal typical human cell, the rate of DNA damage by this mean is about 100 deaminations per day, and that's a huge 100 deaminations per day. Here you see what is the mechanism of deep urination and deamination. Here you see the deep urination, the guanine when there is a problem of the hydrolysis. This is the backbone of the DNA, and after deep urination the base will be missing, because this base it does not have the normal function after getting water. The function of this guanine base is totally changed, so it will not be available for any pairing. So the base will be missing whenever there is a deep urination, and whenever there is a deamination, the cytocine will be converted into the uracil, just because there is only difference between cytocine and uracil is this amine group. When this amine group is removed out of the cytosine, it will be converted into the uracil. So that's why DNA has more chances of these types of mutations. Okay, and we will see that why nature has gone for thymine rather than uracil. Okay, and when uracil is there in the DNA, and you know that the uracil is something that is not normally present in the DNA. So when uracil is present, it will be corrected definitely, because it is not normal part of the DNA. And DNA damage can also be occurred because of the environmental mutagens, and one of the most prolific example is the DNA adducts. These DNA adducts they are formed between the adjacent bases of the double helix. That's why the distortion of the DNA structure occurs. So I will put forward one example in that the example of toxins, aflatoxins. These aflatoxins, they are produced by some of the fungus, and that fungus is present in the corn, in the maize, right? That maize has a fungus, and the fungus that produce a toxin that is known as aflatoxins, ochratoxins, and these toxins can cause damage to the DNA, and it forms DNA adducts, and because of that mutagens it can cause cancer, and they are reported mutagens. And irrespective of the toxins, there is of course the radiations, right? The radiations, the UV light can cause changes in our DNA, and how the mechanism of pyrimidine dimers. Pyrimidine dimers will be made in the formation of the covalent bonds between the adjacent pyrimidine bases, and because of that bonding, replication and transcriptions both are blocked. And when these two processes are blocked, it means there is a blockage of every function of the DNA. So that's why UV is so dangerous. So similarly, the X-rays and related forms of the radiations emitted by the radioactive substances, it may cause the mutations as well, because they can remove electrons from the biological molecules, and when the electrons are removed, again those molecules become functionless. Here you see the thymine dimers, how they are made. Ultraviolet radiation, it contains lots of energy, right? And here you see the thymine dimers. Here the methyls and the nitrogenous and the nitrogen part, they have connections with each other, and this bonding is going to cause the distortion in the structure of the DNA. Thus, because of this distortion, transcription and replication, they are blocked in the DNA structure. So that's why a complete functionless DNA will be the result of UV affected DNA. So this is the oxygen repair mechanism of the DNA damage. Here you see the repair endonuclease will come, and if the damage is present there, it will cut, excised the damaged DNA, and after that DNA polymerase will be there, and it will continue to add nucleotides, and after the nucleotides the DNA ligase will fill up. After the DNA ligase is going to fill up and ligate the adjacent bases, and then the problem is solved. So mismatch repair of the DNA damage, it targets the errors made during the DNA replications. You see, whenever there is a DNA replication, I told you that sometimes the repairs, sometimes the nucleotides, they are not properly attached to their relevant nucleotide. So what will be the further step? The proofreading mechanism. Proofreading will be exonuclease activity. The incorrected base will be replaced by a correct base, but again sometimes that proofreading mechanism, it does not work, right? So what will happen to solve this problem? The mismatch repair system should be able to recognize which of the two DNA strands is newly synthesized. The one of the strand is a template strand, the other strand is a growing strand. So the change, the error, the mistake definitely will be in the newly synthesized strand rather than the template strand, because the template strand is already made, it is already there, and using that template strand, the newly growing strand, the newly synthesized strand will have the error, not the template strand. So that's why. So this was about the mismatch repair of the DNA damage. And so what is the story of uracil, cytosine and thymine? As I told you that for many years it was not clear that why DNA contains thymine instead of the uracil found in the RNA. So now we know that methylation step used to create thymine is energetically expensive. This is one of the reasons that here you see the methylation step. The methylation step, whenever there is a methylation of any kind of molecule, it means that molecule will be more stable as compared to non-methylated molecules. So uracil is less stable and thymine is more stable because of the addition of the methyl group. And the second reason is that, as I told you, the deamination reaction is going to produce a uracil. So if uracil is naturally present in the DNA structure, what will happen if there has been a deamination reaction? So our DNA it would have been impossible to differentiate between original uracil and the uracil that has been developed by the deamination of the cytosine. So that's why nature has gone for the thymine and not for the uracil. So that was the reason why thymine is there in the DNA and not the uracil. So this is the versatile molecular toolkit. Many of these repair proteins, they play additional role in other important activities, and that includes gene transcription, genetic recombination, control of the cell cycle. And this molecular toolkit appears to be used for a variety of DNA related activities. So our next topic is nuclear and cell division, and I hope that this topic has already been covered in our matriculation O and A levels and FSC, but here we will discuss some of the very important points, and you can go through from the book and from the slides as well. So here we have mitosis and cytokinesis. As I told you that mitosis is the nuclear division, and the cytokinesis means it will divide the cytoplasm accordingly after the nuclear division. So prophase, prometaphase, metaphase, anaphase, telophase, these are the 5 phases during the mitosis. And the purpose of the mitosis, as all of you know, that 2 daughter nuclei must receive one copy of duplicated chromosomes, right? So this is the main objective of the mitosis, that the daughter cell must have the equal number of chromosomes. So interphase versus prophase. So interphase versus prophase. Interphase, as all of us know, that this is the phase where the chromatin material is visible, but it is some type of the chromosomes where the chromatin material is visible actually, but it is not clear as it is clear in the prophase. Prophase, you can clearly see the chromatids and the sister chromatids as well, but here you see, you cannot see the chromatids over here. So, and also the esters that are formed, they are visible in the prophase, but they are not visible here in the interphase. So the interphase interphase is the phase where the cell is actually going to prepare for the cell division, and the prophase is the first phase of the M stage, the mitotic stage. And during this prophase, the chromosomes consisting of the two sister chromatids, they are prepared. Then there is a centromere visible, and nucleolus is started disappearing. Here nucleolus is quite visible. And then there are some microtubules, they are going to form mitotic spindle, and here the mitotic spindle is not that much clear. Okay, so the prophase stage is condensation, individual chromosomes are visible, and then also the microtubule organizing center MTOC, where the microtubules are assembled and anchored, that is quite visible. And then mitotic spindle is formed, esters are formed, and centrioles are formed, as I have shown you in the previous slide. So prometaphase, this is the phase where the kinetochore is formed. Kinetochore, what is kinetochore? Kinetochore is a kind of structure that is present here. You see that the kinetochore, it is a central structure, right? And it is the structure where the sister chromatids are joined together. And this kinetochore is very important because it is present in the central point, so a pull will be exerted over here on the kinetochore, and one of the part will go towards this side and the other part will go towards this side. So that's why kinetochore is a kind of balancing position for the chromosomes, and some additional proteins are needed at the centromere. So that's why the structure that have attachment with the pure chromatids to the spindle microtubules. Okay, so this is the sequences, the centromere sequences whose makeup varies from one species to another species, and one of the key element here is that the nuclear envelope is gone, fragmented. Okay, and then comes the metaphase. The chromosomes are aligned together, sister chromatids are tugged towards the opposite poles, and chromatids, they are in a kind of a tug of war between the two equally strong opponents, right? And after the metaphase, we have the anaphase, where the pull will be exerted. This is the image of prophase, this is the prometaphase, and this is the metaphase, and here is the anaphase, and then the telophase. This is the example of a plant cell. This is the micrograph. It shows the mitosis in the cells of onion root, and they are the original cells that were observed through the light microscopy. So anaphase A and anaphase B, these are two parts of the anaphase, but remember that the anaphase is a phase where the pull will be exerted at the opposite poles okay, and anaphase A, chromosomes are pulled the centromere first towards the spindle poles, and the kinetochore microtubules get shorter and shorter because of that pull. And during the anaphase B, the poles themselves move away from each other, as the polar microtubules, they get themselves long. Okay, so depending on the cell type involved, anaphase A and B may take place at the same time, and sometimes B is after the phase A. Here you see this is the spindle pole, and this is the anaphase A, the chromosomes to the pole movement, and this is anaphase B, where you see the pole to pole separation is going on. So telophase is considered as the last phase of the M stage, and during this the poles of the spindle fibers they have arrived, the data chromosomes have arrived, and nucleoli, they start developing, and nuclear envelopes, they are rearranging themselves to form the daughter chromosomes. And during this period, cell duty undergoes cytokinesis, that means the division of the cytoplasm, and equal volume or equal the amount of the cytoplasm is present in the two daughter cells. So that was all about so this was all about the different phases in the M phase, and then there are some terms, some definitions, and I would like you guys to have understanding about all the microtubules. Microtubules, these are the same microtubules that are present in the cytoskeleton. Okay, you can review my lecture on the cytoskeleton and you can see that the microtubules, they are the part of the cytoskeleton, and here the nature utilize these microtubules for a different function. That function is the division of the cells to form fibers and to form ropes that can pull the chromatids apart. So this spindle is necessary for the equal division of the chromosomes in a parental cell into the two daughter cells. Okay, so here the microtubules, they perform a different function than the cytoplasm itself. And ester, it is a cellular structure. It is shaped like a star and it is formed around each centromere during the mitosis. And ester trees, these are composed of again the microtubules, and we will see in a minute in the next slide that how do they look like. The mitotic spindle is responsible for chromosome movement in mitosis. Well, this is the mitotic spindle. It is form of the microtubules, right? And the spindle assembly, there are three steps: spindle assembly and chromosome attachment. And after the attachment, chromosomes must be aligned together and then separate. And the third and the last part is the motor proteins are there to force the chromosomes to move to their respective poles. So these three steps are here, and we will study one step. First, the first step is, here you see the polar microtubules. We have ester microtubules over here. We have spindle poles, and here is the kinetochore. I told you kinetochore is the central point. This kinetochore, and with the kinetochore microtubule, it is attached directly with the spindle pole, with the centrioles. And here the pole will be exerted. These red ones, these are the polar microtubules. These are to maintain this equatorial plate. There is an equatorial plate on which these chromosomes will be aligned together on the same line, and this will be the balancing point, positive with positive and negative. And you see these spindle fibers and these microtubules, again they are the microtubules. They are composed of the tubulin subunits. Tubulin subunits, that is the monomer required to make the microtubules, right? So these were different types of the spindle microtubules. And spindle microtubules, the two ends of each microtubule are chemically different, the -1 and the +1. And during the late prophase, microtubules growth speeds up dramatically, and initiation of the new microtubules at the centromeres, it normally increases. So the contact between the microtubules and the chromosomal kinetochores, it is a kind of possible that the nuclear envelope, when the nuclear envelope is broken down. Okay, so the binding known as the kinetochore microtubules. Here you can see the depolymerization, the polymerization, and again the depolymerization here. These are the tubulin subunits, and these tubulin subunits, they, in the positive, they keep adding, and on the negative they keep removing themselves. So by this process we can see that the polymerization process again is so important because of the recycling. Here the microtubules composed of tubulins. These tubulins, they are recovered at the end of the cell division, and then they, in the daughter cells, they are going to perform the function of the cytoskeleton for the stability of the cells. So polar microtubules, they have different functions. They have direct contact with the polar microtubules from the opposite centrosome. So also the cross-linking between the kinetochores and the kinetochore microtubules, this cross-linking again give the stability to the polar microtubules, and that stability is needed for the proper division of the cell. Okay, so the second point is how the chromosomes are going to align and how they are going to separate from each other. Here you see the tubulin subunits gained and lost. So tubulin subunits, they are making 1, 2, 3, 4. So these are the tubulin subunits. They are getting in here and they are getting out over here. So the positively charged end and the negatively charged end, where the tubulin subunits keep themselves gained and lost, and that is a kind of process that gives a balance effect for the separation of the chromatids. Here the kinetochore microtubules, here the positively charged, here we have negatively charged, and here we have another thing, the motor proteins, that we will discuss in the next point. So here the pull will be exerted on the kinetochore, and the kinetochore have a different microtubules, and these microtubules will pull this chromatid towards this pole, and this chromatid will be pulled towards this chromatid, this centromere. Okay, and here you see when the pull is exerted, the kinetochore is moving this side, the kinetochore microtubule this will be moving this side, and the mitotic centrosome, the centriole pair, is also present in this side. So this is the detail that I have already told you, that during the process of spindle formation and the chromosome alignment and the separations, these are the chromosomes, they appear aligned together and they are separated from each other. You can study this, and I will also share some video with you on these points. So the last point was about the motor proteins and the chromosome movement. So these motor proteins, basically they are present and they play a very active role during the mitosis, because energy is needed for the separation. Whenever there is exertion of the force for the detachment of the chromatids, energy is needed. So that energy is provided by the motor proteins. So that motor proteins, these are present on the kinetochore, and they belong to kinesin family of the proteins, and two such kinesin like motors, they are involved in movement of the chromosomes towards their respective spindle poles. So one is located at the plus end of the kinetochore microtubules, and the other is located at the minus end. So microtubules, they go through depolymerization as well as polymerization during this process. So as a result, we can see that after all these three processes, the chromatids, they are moving towards each other. Here you see all three things, the microtubules here, ester microtubules, then we have motor proteins, they are attached on the kinetochore. This is the kinetochore, and motor proteins are inside, and these motor proteins cause a pull that is needed for the separation of the chromatid. Here you see the bent chromatid, it will be pushed towards the centromere poles. Okay, so this is the miracle of the nature, that in the microscopic cells, all these pulls and push, they are present, and they cause and all this process is just ended in minutes. Whenever the cell divides, it just takes few minutes and the cell is divided through these complicated processes. And this is the slide that shows different parts again. Here, after the M phase is finished, you have two daughter cells waiting for a new life. So these are the mnemonics you can use. And this is I for the interphase, and the prophase, metaphase, anaphase, telophase, and cytokinesis. This is the summary where we can understand, we can remember that what at interphase what happens, and at prophase what happens, metaphase there is an alignment, and at anaphase there is a pull, and the telophase, the complete separation of the daughter nuclei, and then in the cytokinesis, the complete separation of the two daughter cells. So cytokinesis divides the cytoplasm, and it is needed by the daughter cells, and daughter cells later on will be increased in their size, and they will be ready to perform their functions. So in animal cells it is different, and in the plant cells it is different. Here the cleavage furrow will be formed in the animal cells, and the daughter cells will be divided into two different. But in the plant cells there is always a plate, cell plate, a formation of the cell plate, and that will give rise to two different daughter nuclei cells. So that was all about the phases of different cells, and we will have a new part of this chapter in the coming video. Thank you very much.